Before your name was ever spoken into the world, you were moving. Millions of distinct biological entities navigating toward a single destination, the egg. You are the specific, mathematical victor of a massive biological trial where only one could survive. That victory placed you in a dark, enclosed environment, but it was far from empty. Your first understanding of existence was the steady rhythm of your mother's heartbeat. You were formed and protected by an intricate system doing the breathing and carrying for you. You did not drift into existence by accident. You earned your arrival, sustained by an interlocking maternal infrastructure that predates you. That biological infrastructure has an ancient origin. Science identifies a specific woman living tens of thousands of years ago whose cellular energy lineage never stopped. This animated diagram breaks down a human cell to show internal structures called mitochondria. These microscopic engines pull in oxygen molecules and convert them into continuous pulses of energy, driving every physical movement in your body. They are inherited exclusively from the mother. Despite having this powerful internal engine, modern science hit a wall. For decades, researchers tried to study how our bodies fight viruses or repair mutations. But the moment human cells were removed from the body and placed in a lab, they rapidly broke down and died. Doctors could not cure what they could not observe over time. To protect the species, medicine required a stable, unbreakable biological foundation, a testing ground that simply did not exist. In August 1920, a girl named Loretta Pleasant was born in Roanoke, Virginia. Later known as Henrietta, she grew up in a small cabin on land once worked by her enslaved ancestors. She laughed easily, painted her nails red, and eventually married David. This photograph captures the daily reality of communities like Turner Station, Maryland, where Henrietta and David eventually moved. David worked at the Bethlehem steel plant, while Henrietta built a home and raised 5 children. She lived quietly within a marginalized, working-class environment, entirely disconnected from the scientific establishment. In nature, there is a principle called gravitropism. Before a tree can grow upward toward the sun, its root system must first force its way down into the dark, pushing through hard dirt and heavy pressure. The unseen work in the dark dictates the height of the branches. In her early 30s, Henrietta experienced severe pain and bleeding. She sought help at Johns Hopkins, one of the few hospitals treating Black patients at the time. Doctors discovered an aggressive cervical cancer. During her treatments, they cut 2 samples of her tissue. They did not ask her, and she did not know. This microscopic photograph shows what happened to those samples. The blue-stained shapes are Henrietta's cells. Instead of dying within days like every human cell extracted before them, they survived. They divided and divided again, multiplying endlessly to form the first immortal human cell line, named HeLa. This image reveals the immediate shift in medical capability. A scientist is shown observing these cells on a monitor. Because Henrietta's cells did not age or decay, science finally possessed a permanent, stable testing ground. Researchers could now introduce a pathogen to a human cell and track its effects indefinitely. This stability yielded massive, tangible results. This photograph shows mass quantities of the poliomyelitis vaccine. The ability to safely develop, test, and distribute this cure globally relied entirely on the HeLa cell line as its biological baseline. Henrietta Lacks died at 31, completely unaware of what her body had produced. Her unacknowledged physical suffering became the indestructible infrastructure that protected millions of people from disease. Today, that infrastructure supports genetic engineering. Technologies like CRISPR use guide RNA to locate highly specific genetic sequences in our DNA, allowing enzymes to make precise cuts. CRISPR disables a specific gene called BCL11A. Breaking this switch restores fetal hemoglobin, a form of blood present before birth. To achieve this precision, researchers relied on the stable multiplication of HeLa cells to test the cuts and verify the edits were safe. Testing this process in HeLa cells confirmed that the body can be prompted to revert to its own earlier, more resilient genetic blueprints. This mastery shifts the focus of medicine from constructing synthetic cures toward restoring the biological strengths we already possess. This resilience is now being tested against the most hostile and unforgiving environment humanity can reach: outer space. Beyond our atmosphere, human DNA fractures under intense radiation and breaks down in microgravity. To understand how to protect astronauts on long-term missions, scientists send HeLa cells into orbit. They are actively observing how these immortal cellular structures repair their own DNA and regenerate in a vacuum. From the first microscopic race toward the egg to the exploration of the solar system, human life relies on a continuous maternal lineage. Henrietta Lacks exists today as an active, living biological system, her cells providing the essential infrastructure for